This meeting is being recorded.
Hello. So, I'm going to start a new series over Zoom/YouTube because I'm going to upload these videos to YouTube about me explaining science to myself, because I have realized over the years that me teaching helps me understand concepts. That was a common theme that I noticed in my organic chemistry classes, my physics classes. So, everyone's fed up with me teaching them biochemistry, so I'm just going to record these videos and then upload them and then watch them later myself. So I do have a little note PowerPoint thing I want to broadcast. So go ahead and put that up. Okay. Sorry. So this one is going to be all about glycolysis. Okay, so glycolysis is just harnessing the energy from glucose and breaking it down into pyruvate. So just looking at kind of generally an overview of glycolysis, it comes from the Greek word glykos, which means sweet, and lysis, which means loosening. Wonderful. So the breakdown of glucose, I know it comes in a series of reactions, and the first thing to note is that the products of glycolysis include pyruvate, which is then, that's not the end of like the energy making system. Pyruvate is then put through other reactions to expand the amount of energy that is produced. So one molecule of glucose is not entirely energetically used up in one round of glycolysis. Rather, the pyruvate then goes on to different reactions depending on what the body needs to make more energy. We also have ATP, which is adenine triphosphate, from adenine diphosphate, and ADH, which is a reduction of NAD. Pyruvate, like I said, can be further aerobically oxidized, and a precursor. Yeah, okay, we get that. Okay, so has a central role in glucose metabolism, obviously. There we get 2,840 kilojoules per mole from an oxidation of glucose to carbon dioxide, which is not all happening through glycolysis. That's like the complete oxidation of a glucose to carbon dioxide and water. So down here is like just a diagram of the uses of glucose. So it can be stored as glycogen. It can be oxidized through glycolysis to make pyruvate, oxidized via the PP pathway to ribose 5-phosphate. And so I'll make a video about the Pentose phosphate pathway as well later, because that's something I need to review for my exam. And then it can also be used to synthesize polymers. Okay. It happens in almost all species except parasites. So it does not happen in parasites. Sole source of metabolic energy from some cells and some organisms, typically smaller ones. I know pathway with most carbon flux in most cell. Okay, good example of a metabolic pathway under tight regulation. Glycolysis is under very tight regulation, and we'll get to that later. But I know that there's a lot of kind of negative feedback pathways happening later on that inhibit it from proceeding, because you don't want too much energy in the glucose instead of being consistently used, because you want to be able to store it in the form of glycogen. I don't really have to know this, I don't think, but it's the first metabolic pathway to be determined. Okay, blah blah blah. Glycolysis is fermentation, so the anaerobic breakdown of organic nutrients. Okay, glycolysis is anaerobic, so it was made before photosynthesis because it had to be, because photosynthesis is not anaerobic. So the main idea was, how can we extract energy from glucose without the use of air? So it happened in 2 steps. First was to activate it by transferring a couple phosphates to it, and then collecting later energy from the activated glucose. And that's shown here. So primarily we have this preparatory phase, which is to activate the glucose by transferring phosphates to it. And then later we see the whole payoff phase where we collect energy from high energy metabolites. Okay, cool. Like I said, this is using energy, and then we get back energy. So first reaction we see here is the hexokinase reaction, which is what my research is based off of. HKDC1, liver cancer, love her, love hexokinase. So the first phosphorylation of glucose is catalyzed by hexokinase, and a common theme that I'm noticing is typically phosphorylations are catalyzed by different types of kinases. So in prokaryotes, we're seeing that it's glucokinase. In eukaryotes, it's hexokinase. So, and then in addition to that, we also have a magnesium ion, which facilitates the process by stabilizing the negative charge in the transition state. So magnesium, it's just a stabilization. I'm going to think of magnesium as like a mom stabilizing her kids. Magnesium mom, she's like keeping everybody from like being sad and negative and depressed. Love my mom. What molecule provides the phosphoryl group? It's the ATP. So typically when we see hexokinase or different types of kinases like glucokinase in prokaryotes, we see ATP becoming ADP. So it's giving one of the triphosphates to a diphosphate, and one of those is going into whatever molecule we have. In this case, a conversion between glucose and glucose 6-phosphate. Why is it glucose 6-phosphate? Because there's a phosphate on the 6th carbon. Lovely. Yeah, kinases, they transfer phosphoryl groups from ATP to elsewhere. Okay. Yeah, pretty good. Pretty good. That's pretty easy. Phosphohexokinase isomerization. An aldose isomerizes into a ketose. What is an isomerase? An isomerase is something that moves around bonds and electrons, but does not move actual like atoms or functional groups around. Later, we'll see that what does move around functional groups is called mutase. Mutase moves around functional groups. Yeah, so here we just have really like, it's just changing shape. It's becoming a 5-membered ring instead of a 6. So, I mean, there's still like a 6th carbon, it just kind of shortens. So I guess to clarify for myself, essentially what happens is this guy hops up like that. And then the 2 and this guy become connected like that. Okay. So after our hexokinase addition of phosphorylation and after the rearrangement of the 5-membered ring, we also have the first commitment step. Okay, this is what makes the transformation of glucose to pyruvate like happen. Like if this step doesn't happen, it's not like a committed thing. Before this step, it can go down different pathways. After this, we know it's glycolysis. We know it's going to be a pyruvate. There's nowhere else it can go unless there's like a malfunction, which will not be good. So sorry, my family's making noise. What molecule provides the phosphoryl group? ATP, and I can tell you right now it's going to be a kinase, hence phosphofructokinase. Moves ATP phosphoryl groups around. Yes. Irreversible spent product, fructose 1,6-biphosphate is committed to becoming a pyruvate and yielding energy. So like I said, it's committed to glycolysis. PFK1 is negatively regulated by ATP. What does that mean? Negatively regulated by ATP. If there's a lot of ATP, this is what I was talking about earlier about negative inhibition. If there's a lot of ATP, it's going to go and tell PFK, so phosphofructokinase 1, to stop. No mas, no more energy, no more, we have enough. Why? Because if there's a lot of ATP, there's going to be a slower reaction, so we do not want to burn all the glucose. How much energy we have is dependent on how much how much ATP we have. And lots of ATP means lots and lots of energy. So instead of consistently like burning energy, we want to save some of that glucose for when we later need it. Hence it gets stored as glycogen. So hence why this is kind of the commitment, and this is whether I think of this step as the assessment of, do we need to store the glucose or do we need to move forward and make it into ATP energy? But depending on what actually happens, we just have another phosphorylation. So remember that one carbon that we kicked up, that like one that went up and then the 2 attached to the oxygen. Now that one that got kicked up is being phosphorylated. So now we have two of phosphoryl groups being attached to carbons through the same mechanism. So the first one was hexokinase, ATP to ADP, with, again, our lovely mom magnesium regulating all the sadness and negative energy. And now we see phosphofructokinase doing that instead of hexokinase. Okay. Aldolases cleave 6 carbon sugars. So now this part's kind of fun for me. I really like this part, because this is where the numbers start getting tricky, okay? So the aldolase, I believe before we had like an aldolase thing. Yeah, an aldolase isomerizes into a ketose. So aldolase isomerases move around electrons, but now we have our aldolase cutting the whole thingy. Sorry, this is the meeting. moving the whole thing in half, and we have dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, which I remember is DAP and GAP. So DAP became the result from these three guys right here became DAP, and then 4, 5, and 6 carbon became GAP. Okay, so now I have fructose 1,6 biphosphate, and also biphosphate. That bi means that the phosphates, there's two of them, but they're not next to each other. That's what the bi means. So it's on 1 and 6. So the biphosphate now had children. Yes, we have baby little GAP and DAP. Okay, brother and sister. Now, we do not want DAP. DAP is a bad child, and we want to, I guess, convert him into GAP. So we use the triose phosphate isomerase, which converts all of the DAP to GAP. Okay, so through an enzymatic process, all of the DAP becomes GAP. Okay. Now we have this whole issue. It was a little bit confusing for me in the beginning, I'm not gonna lie. Because since all of the DAP and all of the DAP became the GAP, now remember how I labeled like 1, 2, 3 carbon and then 4, 5, 6 carbon? Now they're all in the same GAP thing. So from the derived carbons originally, either carbon 4 or 3 could be like the top one for either GAP or DAP, because remember, like here we had, here it was 1, 2, 3, 4, 5, 6. Now that everybody was converted into GAP, how do we know which one corresponded to which? Well, 1 became like this and this are the same because they have a phosphate group. 2 and 5 are now the same, and then 4 and 3. I hate that this diagram is like backwards. Like, why not just flip it around like that? Whatever. But yeah, that's that. A little bit confusing. Now, after the conversion of all the DAP to GAP is when we start having our little payoff phase. So up until now, we've been investing a lot of energy. So we've invested 2 ATPs so far. Remember, for the 2 phosphorylations, first step with the hexokinase, and then the second step to make it into fructose 1,6 biphosphate with phosphofructokinase, which again is negatively regulated. Nice. So, then we have, like I said, this payoff phase. So glyceraldehyde 3-phosphate dehydrogenase reaction. Dehydrogenase means redox. What is redox? Oxidation is a loss of electrons and a gain in positivity. Reduction is a gain in electrons and more negative, so less positivity. But whenever I see a dehydrogenase, I kind of know, hey, reduction or oxidation, something happening is here, or something with redox. So this is the first energy yielding step in glycolysis because it produces this NADH here. So it's the oxidation of aldehyde with NAD, which gives NADH. So NAD gets reduced to NADH because we're oxidizing the aldehyde. Phosphorylation is a high energy reaction product. Ding, 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 ding. And where does the phosphoryl group come? Typically, when we see that question, we go with ATP because that's what happened in the first and the third step. Now, though, remember that ATP's phosphoryl group little maneuver happened through hexokinase or phosphofructokinase. Now it's happening with inorganic phosphate. Okay, so that's where that comes from. Okay, so let me process what's kind of happening here. So basically, this attaches him himself onto here. Okay, then we have a bisphosphoglycerate because we still have that 1 phosphate here. And this, and now 1, 2, 3, 1 phosphate and 3, so hence it's 1,3 bisphosphoglycerate. Okay. First level substrate phosphorylation. Okay, so 1,3 biphosphoglycerate is a high energy compound that can donate a phosphate group to ADP to make it into ATP. Lovely. The reaction is reversible in the reverse process transfer of phosphate from ATP to phosphoglycerate. Kinases are enzymes that transfer phosphate groups to molecules like ATP. We talked about this. I already said. Whenever you say kinase, there's a phosphoryl moving around with ATP. And look. Our first ATP is made. Very happy. Lots of energy. Love it. Payoff. Big payoff. Love to see it. Lovely. Phosphoglycerate kinase is what's making that happen. Pretty solid. Pretty solid. Not much to say. Conversion of 3-phosphoglycerate to 2-phosphoglycerate. Like I said before, this is just a functional group moving here, like this and this switch. Why? Mutase! I talked about it. Mutase is a type of isomerase that shifts entire functional groups. So remember previously we had the isomerase, and it was an aldose that changed up the electron stuff. Now we're dealing with mutases. Mechanism of the phosphoglycerate mutase reaction. The amino acid that is phosphorylated is histidine. Notice that the substrate from the substrate, phosphate from the substrate, ends up bound to the enzyme at the end of the reaction. Basically, histidine comes in here. Like, I don't know, histidine comes in, okay, takes the phosphoryl group over here, and then brings him over here, and then just drops him off here. Like, histidine's like a traveling pair that's like dropping the phosphate kid off. 2 negative charges in the product are fairly close now, but 2-phosphoglycerate is not a good enough phosphate donor. So now we have to make a better phosphate donor. What happens? Enolase. The loss of phosphate from 2-phosphoglycerate would merely give a secondary alcohol with no further stabilization. So now we have to kind of do a little maneuver here with enolase to make it into an alkene type situation. Where does the phosphoryl group come from here? Phosphoenol pyruvate is where it comes from. So we have an ADP. So remember the whole point of making, like we did this mutase stuff where we switched the phosphate group, but it wasn't a good donor now. So now we got to make it a good donor. Now that we made it a good donor, it can finally donate. So we're now donating the phosphate from the phosphoenol pyruvate into ADP to make it ATP. But loss of phosphate from phosphoenolate yields an enol that tautomerizes into ketone. Tautomerization effectively lowers the concentration of the reaction product and drives the reaction towards ATP formation. Lovely. Okay. So now we have an overall kind of look at this. Okay. So when we cancel everything out, we have a net yield of 2 ATP, 2 pyruvate, and 2 NADH. Okay. Why 2 NADH? Because we only saw 1. For 1 molecule of glucose, the first pathway happens once. And remember, with that whole gap and dap situation that we created, that's 2 molecules, so we had a 6-carbon molecule glucose. We made it into gap and dap, which are both 3. Now each of them go through the payoff phase. So the payoff phase happens twice for 1 molecule of glucose because it was 1, 2, and then we go through 2. So that's why, with the 4, so we ended up using 2 ATP, making 4 ATP, and making 2 NADH. But the net is 2 ATPs because 4 minus 2, 2 NADH and 2 pyruvates. But yeah, so here's where it says that, 4 ATP because you go through the second pathway twice. We used 2 ATP, made 4 ATP, net is 2 ATP, and then, yeah. So that's that. So that's glycolysis. Now we have glycolysis part 2, which is the feeder pathways of glycolysis. So we talked about this. This is just like a practice question. If glucose is labeled at C6 with an isotope of C14, where would it be found in pyruvate? The way that I do this question, on practice, is I draw out the whole glycolysis cycle just because I can then refer back to the diagram that I just drew for further questions, and it helps me visually track where that tagged isotope is. We talked about that. Feeder pathways. So I don't want to talk about all the feeder pathways. I'd rather just talk about the final thing, which is this. Basically, the idea here is that glucose is not the only type of food, sugar, energy source that our body gets in. Sometimes we drink milk, we get lactose. You get sucrose is a different one that's often used. We can get trehalose. We can do D-mannose. These are all like starters that our body ingests. And like we talked about how glucose itself can be like converted down to converted down to pyruvate, but how can the other ones? So we see a kind of feeder pathway of how that happens. So like lactose, with the enzyme lactase gets converted to D-galactose, with UDP galactose converts to UDP glucose, and then glucose 1-phosphate. Now this is the glycolysis cycle again. Then we also have like things like this, where we have fructose 1-phosphate becoming like glyceraldehyde and then dihydroxy, so GAP and DAP kind of thing, where like DAP, we do triose phosphate isomerase to get glyceraldehyde 3-phosphate with glyceraldehyde 3-phosphate. Now you can go through the whole 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenolpyruvate, and pyruvate. So that's that. This one's pretty simple overall. Yeah, so under anaerobic conditions, animals reduce pyruvate to lactate. The acidification of muscle prevents its continuous strenuous work. Lactate can be transported to the liver and converted to glucose there. So once we're done with pyruvate, okay, it can be reduced to lactate. Lactate is then brought to the liver and converted to glucose there. Okay. This is something I'm going to put a star on because I actually completely forgot this, that it goes back into the liver. I knew it was converted back to glucose somehow. I didn't know the mechanism though. Okay. The idea is that like muscle buildup and stuff, like you can't have like lactate just sitting around because it's like acidifying the muscle it's sitting in, and that's why like you can feel it like uncomfortable when you're like doing strenuous exercise, or me frankly doing any exercise. Since I'm super out of shape right now, I get a buildup of lactate, which makes your muscles burn. Hence why it's like your body then's like, all right, chill off, like let's cool down, and the lactate gets sent off to the liver to make more glucose. Under anaerobic conditions, yeast ferments glucose to ethanol. Well, not in this diagram, it doesn't really show glucose, but we know that glucose becomes pyruvate. Pyruvate is then put through pyruvate decarboxylase, which removes the carbon as a carbon gas. Now we have a 2-carbon acetaldehyde. We have alcohol dehydrogenase, which is a reduction because it's redox, because it's dehydrogenase, and it becomes ethanol. Again, we see magnesium in pyruvate decarboxylase and zinc and NAD in dehydrogenase. So oops, yeah, that's that. Okay, I think I'll just review this again for my peace of mind. All right, pentose phosphate pathway. The main goals are to produce NADPH for anabolic reactions. Anabolic means building up a reaction. So we're at glucose 6-phosphate. Okay, so remember how I said that like the first commitment step was the conversion between fructose 6-phosphate and fructose 1,6-biphosphate. So before fructose 6-phosphate is the reaction of glucose 6-phosphate with the phosphohexose isomerase. So when it doesn't go down, when glucose doesn't go down the pathway of glycolysis, it could also go down the pentose phosphate pathway after that initial hexokinase reaction. That adds a phosphate to glucose. So, and that's where our starting point is. Same first step as glycolysis. So then we're going to go down here. We're going to have a little cycle of reduction with NAD and NADPH. So we're yielding an NADPH, happens again, NADPH. And then at this point, when we make the ribulose 5-phosphate, remember, so it was 6-phosphogluconate, and now we're at ribulose 5-phosphate. Where did that carbon go? It got lost as carbon dioxide. We can now go back here through a transcatolase, transaldolase reaction. Transaldolase just kind of rearranges it and adds another carbon to it. And so we kind of just go back here and then go down. Then we have a ribose 5-phosphate, which then gets converted to a nucleotide coenzyme DNA RNA material. So if we're not making pyruvate through glycolysis, we can still use glucose to make nucleotide coenzymes, DNA, and RNA. NADPH regulates the glycolysis versus pentose phosphate pathway. When there's too much NADPH sensed, it stops the production. Hence, NADPH would inhibit the dehydrogenase reaction and not make more NADPH from NADP. So again, if there's too much, it's going to tell, hey, like right here, it's going to be like, hey, stop this, because we're sensing too much NADPH, so stop this reaction here, this dehydrogenase reaction. So, and I should probably include here that this, I'll do it in the red, that this and this is a dehydrogenase because it's redox. That's so bad handwriting. I'll come back to that. Okay, we talked about that. And yeah, so glycolysis and pentose phosphate pathways and the feeder pathways are different ways of using glucose. Again, glycolysis makes energy out of it, makes it into pyruvate. Pyruvate then goes and makes more energy. Pentose phosphate pathway makes it into a something, the ribulose 5-phosphate, which can then be used for nucleotides, DNA, and RNA material. Thanks for watching.